Oh, there you go. Great. Thank you. So it's probably maybe the first time that you've had a plant person speaking, but I'm actually going to talk about transcription, which everybody should care about. But I have to say that for my PhD, I sequenced the yeast mitochondrial genome. And we discovered that TGA codes for tryptophan in yeast mitochondria. And we got a little tiny article in the Times about it. And then when it was discovered that humans also use TGA to code for tryptophan, it was big news. So when I was thinking about a postdoc, I thought, well, there's more people studying yeast genes than there are yeast genes. And in plants, almost anything that I could discover might have a practical application. So I guess I consider my work in Pasteur's quadrant, the quadrant where basic research could have a potential application. And so some of our discoveries are actually patented, licensed by companies that want to get plants to grow better on nitrogen, which is the key rate-limiting element in plant growth. But in studying the basic principles, today I'm really going to focus on a new mechanism of transcription that we discovered that transcends plants. So that's the bottom line. All right, and this work is done in my lab, and I would say everybody in my lab has some computational experience and coding ability. But we do, I know that there's not really a pointer. Can you see this? Yeah. So, but we do a lot of our network modeling, especially the machine learning, in collaboration with Courant. And I'm going to tell you about my work with Dennis Shasha and some machine learning work that he supervised along with Jan LeCun. Okay. So I'm going to tell you today about time. Now, the idea of systems biology is that you start in the old biology, you'd have a hypothesis that you do an experiment, you test it, and then you'd move on. In the era of systems biology, you have a problem that you want to study, but you don't necessarily have a hypothesis. So we want to study how do plants use nitrogen, and so we generate a lot of data, and through the data analysis and network analysis of the data, we actually generate the hypothesis that we want to test. And then once you test the hypothesis and validate it on a genome-wide scale, it refuels this cycle. So today I'm going to tell you that our big data is we learn regulatory networks from time series data. And then the idea of systems biology is not just to take the data that you know and make a model that explains the data, but to make a model that can predict how the system will react at some untested time point. And so I'm going to tell you today about doing just that. And then once we develop hypotheses, we want to test them. And so we perturb, in our case, the transcriptional networks and monitor them in real time. So our validation studies of transcription is in real time. Okay, so those are the two halves. One is modeling, one is experimenting, but every time you experiment, you refuel the model. Okay. All right, and I'm going to tell you that from this, even though we did not start out with a hypothesis, we've uncovered a new hypothesis and new biology. And I'm going to tell you a bit more about that. Somehow I've lost my pointer. And I especially want to make the case for the new biology that we've discovered, because a lot of people say, oh, systems biology, it's just a bunch of networks and can't make any sense out of them. Well, the networks actually helped us, with biological intuition, learn some new biology. So you can't just be a computer scientist and look at networks and discover things. You still have to have biological knowledge. And so part of what we do in our lab is we host a website called the Virtual Plant that enables biologists like me to mine my own data in a software platform that's kind of like a video game. You upload a gene list and you could press a few buttons and get a network. And then you use your biological intuition to figure out what are the subnetworks that are worth pursuing experimentally. And I'll give you a couple of examples. So today I'm going to talk about time. And one of the postdocs in my lab wrote a nice review in TIGS about looking at transcription in time. And the published work spans everything from milliseconds up to minutes. So in milliseconds, transcription factors are looking for targets. Within seconds to minutes, they transcribe genes. And those genes encode proteins that interact. And those networks then lead to changes in organismal responses. Now, many of you know that there are very elegant single molecule studies that can study transcription factor target interactions within milliseconds. And as those beautiful studies, they actually use single reporter genes. And so they're monitoring expression of that reporter gene for these studies, where they're studying, how quickly does a transcription factor come on and go off. Very beautiful studies. The only caveat is you're studying a single gene. So you can't actually derive genome-wide data from those studies. So you study a mechanism of a gene or multiple genes, one gene at a time. And so what I'm going to tell you today, and so I think I'm not putting these studies down. I'm just saying that it's difficult to get the biological message. You'll get a mechanistic message of what's happening of a transcription factor and a target. And what I'm going to try to tell you today is we've gotten a mechanistic view of what's happening with transcription factor transient binding to a target, but we've also figured out the biological relevance. So in our case, I'm going to tell you about a system, a cell-based system that we've developed where we can control TF entry into the nucleus and monitor TF binding over time. We could monitor gene regulation over time. And then we take the data, the time-based data, and build regulatory networks that are time-driven. And from that, the main story I'm going to tell you today is a story about hit-and-run transcription, where a transcription factor comes, binds to a gene transiently, activates its transcription, and that transcription stays on even after the TF has gone. That's why it's called hit-and-run. And I'm going to tell you that this hit-and-run mechanism of transcription enables plants to respond rapidly to an environmental signal, in our case, nitrogen. And so that a single TF, by going from gene to gene to gene, think of it like a bee and a flower, can propagate the signal as a single TF. So it's almost like a TF as a catalyst. It doesn't have to stay there for the gene to stay on. That's the bottom line. All right, so the biological context of our motivation for studying, so there's very beautiful nitrogen studies in yeast. And as a matter of fact, when I went into plants, I was inspired by Jerry Fink's work on amino acid sensing in yeast. But I felt that if we started studying nitrogen and amino acid sensing in plants, it might have a practical application. And so the motivation is that in plants, nitrogen is the rate-limiting element for plant growth. That's why nitrogen is the main component of fertilizers. But unfortunately, crops only use about 70% of the nitrogen that they're given. The rest of it goes back into the environment. So between making the fertilizer via the Haber-Bosch reaction and its release into the environment, it's generating a tremendous amount of greenhouse gases and algal blooms. The other thing that is, if I showed you a map of the world, the only part of the world that has the problem with this over-fertilization is the US in the Midwest. And if you look at most of the world, it's very low nitrogen and they can't grow crops or plants. So we really want to increase nitrogen use efficiency, not just to mitigate pollution, but also to deal with worldwide crop growth. Okay, so, but we don't work on crops directly. We work on Arabidopsis, which is the model. It's the Drosophila of the plant world. Its genome was sequenced back in 2000, actually before the human genome. And it's a data-rich species. There's a lot of data on protein interactions, et cetera, you'll see. But we use it as a tool, and then we use it to transfer network knowledge to crops. And I'm not going to talk about this part today, but part of our virtual plant software platform enables you to go from Arabidopsis to corn or rice or these other crop genomes and transfer information. For example, one nitrogen network in maize that we made by correlation of gene expression, 5,000 genes, half of them had no annotation. And yet when we imported the knowledge from Arabidopsis, as even getting annotation is important. So that's the motivation. In terms of nitrogen sensing, and I'm going to show you how we use time series data, plants don't just like sit in the soil and wait for nitrogen to come to them. They can actually sense it, and plants can actually grow lateral roots in the direction of nitrogen-rich patches in the soil. And plant meristems can form post-embryonically, so they actually change their development in response to nutrient availability. You know, no matter how much I eat, I'm going to be the same size, maybe wider. But it's the root foraging is an amazing ability for plants to grow lateral roots. In our lab, and I'm not going to tell you about it, we take a single plant and split its roots between nitrogen and no nitrogen, and the one on nitrogen goes crazy. And so we did that, and we're mapping genes involved in root foraging. So the signal for that is nitrate. But it turns out that nitrogen assimilation, so assimilation of inorganic nitrate into glutamine, the nitrogen donor in basically all biosynthetic reactions, amino acids, DNA, et cetera. It's the most energy-intensive pathway, biochemical pathway, uses up about equivalent of 12 ATPs. So once plants have enough glutamine, glutamine acts as a signal to repress further assimilation, repress root foraging, but to also induce the synthesis of amino acids like glutamine, which it uses for storage. So plants can sense both external nitrogen signals and internal nitrogen signals. And so I'm going to tell you about how we use time series data in response to these nitrogen treatments to come up with some new biology related to nitrogen signaling. I'm going to tell you very briefly how we've discovered that nitrogen controls the circadian clock using a network approach. The main thing I'm going to tell you about is how we've discovered this new mode of transcription called hit and run involved in nitrogen signaling. And I'm not going to have time, but we also have studied how plants forage for nitrogen in their roots using the split root system that I alluded to earlier. All right, so how can networks lead us to these new biological insights? So this is like bringing coals to Newcastle, but networks are made up of nodes and edges that connect them. And there are social networks, there could be city networks, and there are gene networks, which is why I'm here today. But the networks tell us about how genes function together, and network properties give us clues to how the system can react. So in this view of these city networks, the nodes are cities, and they're a map of the US. And on the left, this is a random network where the edges connecting the cities are highways. And pretty much every node has the same number of connections. But if you look at this map here, which is called a scale-free network, the red nodes are hubs, which are, they have many connections. And so there's few nodes with many connections and most nodes with few connections. And this scale-free network, it gives the system robustness. So for example, if I were to fracture any random node in the random network, the system would fail. But if you fracture a random node in a scale-free network, the system continues to operate because of the hubs. So it turns out that most biological networks are scale-free, which is why we can sustain mutations. But if you mutate a hub, then the system collapses. So our goal is to create networks, identify the hubs, and attack them, because transcription factors in general are usually part of multi-gene families. Making a mutation in one of them usually doesn't do the job. So our perturbation system for inducing a TF to enter a nucleus and see what it does has really paid off to figure out what the TFs do. So I'm going to show you one example of a network. And this network we created we took some nitrogen-treated plants and we did transcriptomics, and we identified about 800 some odd genes whose expression, transcription, responded to the nitrogen treatment. And we treated either with nitrate or glutamine. But then we asked, okay, we have a list of 800 genes, so what do we do with that? So we built something called the multi-network. So we took all available data published or in databases that connects two genes in Arabidopsis. And that could be based on if two genes are in a metabolic pathway, if a transcription factor regulates a target gene. There are some databases called AGRIS where that information is stored. Protein-protein interaction data is available for plants, microRNA, RNA data. And there's even in this network a text mining edge. There's a program called GeneWays by Andrei Retsky, who's I think at Chicago. If two genes are in a sentence and the word bind, interact, or activate is in that sentence, the program will draw an edge between those two genes. So you actually don't have to read anymore, which is always. So what we did was the multi-network has 20,000 genes and 180,000 interactions. So it's this big hairball, but I call it the encyclopedia. It's like Encyclopedia Britannica. And if you query that hairball, you can now extract the genes that are regulated by nitrogen and learn all of their interactions. And roughly half of the nitrogen-regulated genes were connected by at least one of those types of interactions, which tells you that these genes are working together. It's actually a pretty surprising number. All right, and so now we take advantage of network properties. It turns out the top two hubs of that network, and here's where the biological intuition comes in. One is CCA1 and the other is BZIP1, which I'm going to tell you more about today. But CCA1 is a MIB transcription factor that regulates the circadian clock. And here it was as the top hub of a nitrogen-regulated gene network. So we derived the hypothesis that CCA1 is regulated by nitrogen. Actually, that's what the data told us. But then the hypothesis is that nitrogen regulates the circadian clock. So to test this, we took the CCA1 promoter, which is itself circadian regulated, and it's fused to luciferase. And in collaboration with Rob McClung, we could see if we grow these plants in continuous light, we see circadian expression of the CCA1 gene, as expected. But if we gave the plants pulses of either nitrate or glutamine in this case, we could either phase delay or phase advance the clock. So you phase shift it by 2 hours. So this is basically a validation. People knew that the clock was regulating genes in the nitrogen assimilation pathway, but the new knowledge is that the products of nitrogen assimilation feedback regulate the clock. And so nutrient regulation of the clock was a new principle in biology. So as beautiful as this example was, it really was based on static data. And if we go back to the analogy of the US, this now shows flights coming into the US from Europe. You could see that networks are dynamic. And you could see even that there are some hubs that are forming over time that you would not know if you only looked at one static picture or if you even looked at the end of this picture. And so it turns out that time series data is because time moves forward, time series data are a valuable source to make predictive networks. So we collected nitrogen response data over time. So this is a heat map of transcriptome of plants treated from 5 minutes to 120 minutes. And there are genes that are either induced or repressed in response to nitrogen treatment. And interestingly, if you take each of the time points and say, tell me when a gene is first activated, and then if you look for overrepresented cis elements in those classes of genes, you can see that there's sets of cis elements that come up at each time point. So this is telling us that there's a regulatory cascade of transcription factors that come up over time to induce the sets of genes that become activated at 5 minutes or 10 minutes or 15 minutes, et cetera. And so now I'm going to tell you how we used this time series data to generate the regulatory networks that underlie this cis element analysis. And this is the work that was done by Denis Shasha at Courant with a student, Piotr Minorsky, and by a very talented postdoc in my lab at the time, Gab Crook, who was a Marie Curie fellow. So I'm going to tell you how we learned regulatory networks from time and used them to predict. So prior to this work, the earliest time point that anybody had tried to treat plants with nitrogen was 20 minutes. And Gab went back to start at 3 minutes. And I said, why 3 minutes? He said, well, it was the fastest I could transfer the plants from one media to the other and collect it. But what he found was that if you plot out the percent new knowledge prior to the 20 minutes, there was a whole host of genes that became active very early on, and some of them transiently, and so they were missed at the longer time points. And if you see here, these are the nitrogen use genes. These are genes that, for example, a nitrate transporter, it comes up compared, so this is KCl control versus KNO3. This gene starts to get activated about 20 minutes, and this nitrate reductase gene at about 15 minutes. And so we were really interested in what are the genes that come on earlier. And it turns out that a lot of them, the early genes, were transcription factors. And so we asked ourselves, is there a causal relationship between these early regulated transcription factors and the downstream genes in the targets? And so with Denis, we used a method called state space analysis. And so I asked Denis, I was like, okay, explain state space analysis. And he drew those balls for me. So that's like a computer scientist version of state space analysis. And I'll give you the biologist version in a second. So these are Y, it's Y, T, T plus 1, T plus 2. Those are our time points. And basically, state space modeling uses Bayesian and Markovian approaches in which each gene expression value at time T plus 1 is dependent only on the state of all genes at time T. They used a noise mitigation approach. So Y is the observed values, and this is some kind of factor that they use to deal with noise. That's all I know. And basically, in the end, the goal is to learn this dynamic model where you can explain the expression of a gene at time T plus 1 based on the expression of transcription factors at the previous time point. So basically, you're using the RNA levels as a proxy for the amount of protein or activity of a transcription factor. Right, Marion's squeamish about that. And so here's my version of state space. So basically, here's our transcriptome data at time T, T plus 1, T plus 2. And we're trying to learn this F function. And this F function basically takes the expression levels of the TFs at time T, and they can either act in a positive or a negative way to predict the expression of a target gene at the next time point. And you do this, you learn iteratively. And the equation looks something like this. This is a simplified version. Basically, you know the expression of gene I at time t, and you know the expression of a set of transcription factors at time t. It includes an mRNA decay rate and Gaussian noise, and you try to learn a dynamic F function which shows you the set of TFs that can explain the change in expression of this gene from time t to time t plus 1. And when you run this, you get a matrix that says, this is the regulated gene, and these are all the TFs that can explain its expression at the next time point. So computer scientists do this, and the way they test their model is to, instead of training on all of the data points, you leave out one data point, and you actually have the data, you put it in your back pocket. And so they trained the data from the 0 to 15-minute time point and left the 20-minute data in their back pocket. And if my algorithm is correct, I should be able to predict expression of a gene in my network at the 20-minute time point. And it turns out when we use state space, we were 74% correct in predicting whether a gene would go up or down at the 20-minute time point. We couldn't predict the magnitude of change. And I said to Dennis, well, you know, if I saw that a gene went up from 15 to 20 minutes, or 9 to 15, I'd say it's going to go up at 20. And that's called a trend forecast test. And that's just as good as tossing a coin. So the learning was kind of remarkable because it's a really small data set. You know, that's not that many time points. But Dennis thinks it's because of the noise mitigation approach. So that's the good news. The other good news is that this allowed us to make this dynamic network of all the genes in the nitrogen assimilation pathway and the transcription factors that potentially regulate them over time. And we could use the network structure to say, okay, there's the hub, it's controlling most of the genes. But the scary thing is there's 38 transcription factors, and they're all controlling each other. And in Arabidopsis, it's not so easy to make mutants in 38 like it is in yeast. So this prompted us to try to develop a high throughput system to test transcription factor function, because we wanted to validate the TF target interactions and also use the validated data to refine the model. So we now developed a system to perturb regulatory networks in real time, to track transcription in real time, and that's what led us to discover this hit-and-run transcription, which is the next part that I'll tell you about. Okay, so the way the system works is it's called Target for transient assay reporting genome-wide effects of transcription factors. And basically, we can control a TF entry into the nucleus. I'll show you that in a minute. And then we can look at early events and late events and identify the direct targets, either by transcription factor binding or by transcription factor regulation of the target gene. So this is a functional assay. So the assay system works like this. So we take roots from whole plants and we dissociate their cells into individual cells. And we transfect those root cells with the plasmid that contains an RFP reporter gene driven by a constitutive promoter. So any cell that gets this plasmid will fluoresce red. And then on the same plasmid, we overexpress a transcription factor. And the transcription factor has a small fusion protein to it, the glucocorticoid receptor domain. And what that does is when the protein is expressed, this glucocorticoid receptor domain binds to HSP90. And even though you're expressing the protein, it holds it in the cytoplasm. So then you could treat with dexamethasone, which disrupts the HSP90 binding to your fusion protein, and your TF goes into the nucleus. And so you can precisely make the TF go into the nucleus and then look at your watch, and determine what happens over time. That's what the system is about. So we could determine what genes the TF is bound to because we can use an antibody against the glucocorticoid receptor domain to pull down the DNA fragments that are bound to the TF. So those are direct targets. But the beauty of the system is we could also identify genes that are regulated by the TF. And you probably all know that TF binding is a really poor indicator of the genes that are regulated by a TF. And so TF regulation is a functional assay of whether or not a transcription factor is able to recruit Pol2 and cause changes in RNA. And so by doing this step in the presence of cyclohexamide, where we block translation of, let's say, a message encodes another transcription factor, if we block translation of those primary messengers, then we could say that all the genes that got activated are due to our original transcription factor and not to a downstream TF. And because everybody hates cyclohexamide, we did a filter, which is we only took the genes that are DEX regulated either in the presence or the absence of cyclohexamide. So you can't say it's some funky cyclohexamide thing. So we're missing data, we're missing feed forward, feedback loops, but this will tell you it's a direct target and it's regulated. Okay, so the data looks like this for BZIP1, and this is kind of an important point. Everybody talks about transcription factors as activators or repressors. As far as we could see, every TF that we've tested could be either. So, and I think I'm going to tell you it's because of the context of the targets. So in this case, the BZIP1 we treat with dexamethasone to induce BZIP1. So minus and plus, so the 2 plus dexamethasones are this induced genes, and these 2 are repressed genes by BZIP1. And we also, I didn't make a big deal of this, but we also treat in the presence or absence of the nitrogen signal that the TF transduces. And so that you could see, and I'm not going to go into this, but there are genes that are regulated specifically by the interaction of the TF and nitrogen, and we do that using 2AA NOVA. So you could find out genes that are responsive to the TF, to nitrogen, or to the combination of the two. Okay. And then separately, we did ChIP-seq to get the bound genes. And so here's the input DNA, and the blue tells us the pieces of DNA that were pulled down in our ChIP. And when you line those sequences back up to the genome, those hills are where the TF is bound. Actually, the valleys are where it's bound. And because of the way the sequencing reads go around the TF, you can see exactly where it's bound. So we had these 2 data sets, the binding genome-wide and the regulation genome-wide. And of course, we wanted to compare them. Now, what I didn't tell you is both of these data sets came from the same cells. A lot of time when you look at ChIP-seq data and expression data, they came from different tissues, different experiments. So we expected a really good overlap of bound and regulated genes from this data set. Okay, so here was the surprise. So we found about 400 genes that were bound to BZIP1 but were not regulated. And that wasn't a surprise because we know binding is a poor indicator of regulation. We found 120 genes that were bound and regulated, and everybody gets excited about this class. It's like the gold standard. If it's bound and it's regulated, go for it. And then everybody finds regulated, not bound, and they usually throw them away and they say it's a secondary target. But we knew they were primary targets because we did that cyclohexamide thing. So the other, and I remember going home that weekend and my postdocs were all excited about the 120 genes. And I kept thinking, what's going on with those others? And it turns out when we looked at these genes that are regulated but not bound, they were all these interesting genes involved in nitrogen signaling, some of which we knew based on Go terms. And so we came up with a hypothesis that their regulation is dependent on getting the TF into the nucleus. So we hypothesized that they were transiently bound. But I won't go into details. These putative transient targets are enriched in genes that respond to nitrogen in our cell-based system. They're enriched in genes that show a nitrogen by transcription factor interaction. And they're also uniquely enriched in genes that respond early and transiently to nitrogen treatments in whole roots. So if you look at these classes, so class 1 is we call it poised. The TF is bound. Class 2 is bound and regulated, and it's either induced or repressed. That's what the A and the B are. And what we call transient are the ones that are regulated and not bound, either induced or repressed. And so the induced genes include all of these nitrogen and early response genes, including ones like these nitrate receptors and transporters. So we knew that this transient class was important and usually overlooked. And so to prove that they were transiently bound, we used the target system, but now we did time series ChIP-seq. And interestingly, we're talking about 10,000 cells in each sample. We were able to get ChIP signal after 1 minute of TF nuclear entry. So it's not milliseconds, but it's 1 minute. And so if you see on the top, so NLP3 is a famous gene involved in nitrogen signaling. On the left, the y-axis is the ChIP-seq reads. The x-axis is this specific gene. It's transcription start site, plus 500, minus 500. So you could see that after 1 minute, we find BZIP binding, 5 minutes, and then by 30 and 60, it's gone. So this was proof that, and we had this for a number of these early response genes. But we knew that we're probably missing genes because ChIP-seq is really bad at false negatives. You miss things because they're not stably bound. So we became intrigued by finding other ways to see if a transcription factor is transiently bound to a gene. And there's this really old paper, you know, in my lab when we say it's 1980s, you know, this is 2001, but that was a while ago, where they used a DNA methylase fused to a transcription factor. And the idea is that the DNA methylase will, if it touches down on DNA, it methylates the adenines, and then even if it goes away, it basically left that footprint, which is forever. Well, I don't know, forever. And so we adapted the system by putting on the GR tag so that we could control entry into the nucleus. And that's important because you don't really want your adenines methylated. So people tried to do this in Drosophila and C. elegans, and they really didn't like having their adenines methylated as they were developing. So because this is a transient system, we could get around that problem. All right, so we have our GR BZIP1 DAM fusion. And the idea is that this fusion protein, when BZIP1 binds to its recognition site, the DAM methylase will methylate adenines. And plants don't have adenine methylase, so there's actually no background. And so then you can look at a target gene by looking, you basically digest with DPN1 when it's G, A, T, C, and when the A is methylated, DPN1 will cut. So you basically just cut your DNA and then map it back to the genome to find the promoters that were touched by your TF, even transiently. And so when we did that, here's our we got about, oh, I don't have the number up there. I think we got about 1,000 genes that were touched by the GR BZIP DAM fusion. And when we intersected that list with the genes that we identified by our ChIP and expression studies, the important point is we found that DNA methylation of genes that are in the transient class, this is another level of proof that those genes were touched by our TF at some point. And so we're perfecting this because we want to deal with background better. But these early results were very convincing that this was another method to prove and capture transiently bound genes. All right, so the last part of the story is, okay, so we know that there's transient binding of a TF. But in our assay, our time point that we collected our transcriptome data was 5 hours after TF nuclear import. And we wanted to know were those genes actively transcribed at a time point when the TF was no longer bound? So the genes needed to be activated by that TF, but were they the result of a gene that becomes activated and stays high because it's a stable message? Or is that gene actually being actively transcribed at that 5-hour time point when the TF is not bound? And so to do this, we collaborated with David Gresham at NYU, who studies yeast, Marion. And we used 4-thiouracil. So it's a thiol substituted uracil that becomes incorporated into UTPs. And so you could use it to label newly synthesized messenger RNAs. And so we incorporated the 4TU into our target system. And so because if now a newly synthesized message is made, it's incorporating the 4TU. And we can distinguish de novo RNA from pre-existing. And we do that using an affinity capture of the newly synthesized message. And so with 4TU labeling, we do 4TU labeling with our BZIP TF. Our control is empty vector. And we look at genes that we capture that are either activated or repressed by BZIP1. And so that means these genes are they're BZIP1 dependent, so they need BZIP1 to be transcribed or repressed, and they're actively transcribed. And so if you think about it in this way, we do DEX-induced TF nuclear import, and here we're labeling at the 5-hour time point with 4TU, whereas the steady state RNA data that we had from our other study, it could be that the message was made initially when BZIP1 was touching the gene, but that is just hanging around because it's stable. And so when we compared our 4TU labeled RNAs that are made by BZIP and actively transcribed, the important point is when we compare them to the poised, stable, and transient, that the transient genes are actively transcribed at a significant level. So that's the overlap between the genes. And so the bottom line is that those genes are transcribed at a time point when the TF is no longer bound. So I started thinking about all this data, and I had all these names for it. I had an airplane. There's something called, there's an airplane maneuver, which is like touch and go. And I was getting fanciful. And then I actually found a paper back from 1981 88 by Walter Schaffner. And he hypothesized that there's something called hit and run transcription, where a TF comes in and hits to activate and initiate a Pol2 complex, but then it runs, but that transcription continues after the TF dissociates. So he hypothesized this. And I have to say, it was also proven on a single gene level. Remember I told you about those single reporter genes when I started the talk? And so the mechanism exists. It exists in animals, not just plants. And single gene studies have reported it. But our genome-wide data has provided insights into the mechanism that I think you couldn't have gotten from single gene studies. And that is as follows. So the question is, why does transcription continue after the TF is gone? The hypothesis is that the hit somehow recruits TF partners to continue the transcription after the initial TF is gone. That was hypothesized. And when we look at our genome-wide data of our transient targets, we find that the transient targets are enriched in the BZIP1 binding element, which is known, but they are uniquely enriched in partner elements as a group compared to the stably bound or the bound and not regulated. And so here's our three classes: poised, stable, and transient. And because they each have gene groups, we now can do cis element enrichment studies. And we find the known hybrid AGC-T box, that's the known BZIP binding elements, is highly enriched, especially in the transiently bound genes. But only the transiently bound genes have these partner cis elements. And this shows the cis element enrichment. So the blue is more enriched, so it gives us a list of putative partners. So this is the kind of insight that you would not have gotten if you were looking at a single gene that was a hit and run target. But the other thing that tells us, and this is really pretty important, is that a single TF can act either in a stable manner or in a transient manner based on the cis context of the gene. So it's not like a hit and run. So in theory, this could be any TF. And I'm going to show you a slide later that has, you all know probably in your fields, there is a very small overlap between TF bound and TF regulated genes across yeast, C. elegans, flies, humans. And everybody's throwing out the regulated not bound because they can't distinguish primary from secondary targets. And I'm positing that a lot of data is getting thrown away and that there's probably transiently bound TFs depending on the context of the cis element. Okay, so last. I keep saying last, but now I really am last. The question is, how are the partners recruited? So we hypothesized in a nice paper in Bioassays that potentially the TF hit could recruit the histone acetyltransferase that would modify chromatin, open it up so the partners could come in. And it turns out that for BZIP1, BZIP1 makes a dimer with BZIP11. And BZIP11 is known to interact with the histone acetyltransferase, which is why we made this hypothesis. And so that would open the chromatin so that the partner TF could come in and lead continued transcription. And in our case, because we know the cis elements, it turns out CCA1 is probably one of the partners for BZIP1 in this hit and run. And then interestingly, we inspired another group to use our terminology, and they had been scratching their head about this result. So there's a heat shock transcription factor called HSF2A2, and basically, in response to a heat shock, the TF binds, that's the red line, so there's binding, but you could see it's transient. But what's not transient is that it initiates chromatin modification, which persists after the TF is no longer bound. And so this is another example now, after ours, about transient TF binding initiating events that persist after the TF is gone. And so I guess the end is that both of the stories, the nitrogen and the temperature, are environmental signals. And so this could be a rapid way for organisms like plants that are, the difference between us and plants is they're stuck in their environment. They have to deal with it, as you say to your kids, deal with it. So they have to deal with their environment, and this is a rapid way to do it. And so basically the perspective is, if you go home, this is what I'd like you to remember: Tom Sawyer. So basically the catalyst TF, BZIP1, is Tom Sawyer. He starts painting the fence, and then he recruits his friends to help him, and he goes away. And when he recruits his friend, we know about pioneer TFs that open up chromatin. That's one model I just told you about. But another model I'd like to propose is that this allows a TF to be a catalyst. So I can go from one gene to another, like a bee to each flower, initiating transcription, but I don't have to stay there. I let my buddies do the work. And I guess I said to you earlier that there's a very, very, very low intersect of bound versus regulated targets across plants, animals, and yeast. And so I'm proposing that hit and run transcription, which we know does occur in animals, is more widespread than we think. And so I'll just end by saying that the work was done in my lab by some very talented postdocs. I already mentioned Gabrielle Crook. Joanne Doherty did the work on the 4TU. And I think Ying and Crantley did most of the bioinformatic analysis for the hit and run story. And so I think I'll stop. I'm on my yellow light and take questions.